Hi, and welcome to my poster, Spatial Distancing, Investigation of a Defense Mechanism for Pathogen Immune Evasion. So in this poster, I will first present the pathogen immune evasion mechanism that we've come up with, and then two different modeling approaches to simulate such a mechanism. So in our work, we are interested in modeling the interactions between on one side the pathogen and on the other side the immune system. And as experiments, what we can do is a whole blood infection assay, for instance, where we take human blood that we infect with the pathogen, and for different conditions, we can look at the system state. And what we see here is that for Candida albicans, which is a commensal fungus, it is mostly uptaken by neutrophil, a little bit by the monocytes, but what is interesting here for us is that there is always a portion of Candida albicans, of the pathogen, that remains free in the system, which means that the pathogen can escape the immune system. And the question is, how can it do this? And to answer this question, we first need to take a step back and look at how the immune system acts as a barrier against pathogens during infection. So here I'm focusing on two different ways. The first one is the extracellular killing via antimicrobial peptides, AMPs. So basically the neutrophil is able to secrete some small molecules that are harmful for the pathogen and would kill it. And the second way is the phagocytosis, and this can be done either spontaneously by the immune cells or after the labeling of the pathogen via small molecules, opsonins. So what we can observe here is that the interactions between host molecules and the pathogen are crucial in removing the pathogen during infection. And based on this observation, we come up with a mechanism. So we have a pathogenic cell surrounded by antimicrobial peptide, and the idea is that the pathogenic cell can secrete its own molecule, defensive molecule, and this molecule will then bind to this antimicrobial peptide, forming a complex. And then the formation of complex would then happen in the vicinity of the pathogenic cell, and due to the gradient, just diffuse away from the cell. And in this way, the antimicrobial peptide are basically transported away from the pathogen, and thus increasing the chance of survival of the pathogen. And we refer to this mechanism as spatial distancing due to this transport of antimicrobial peptide away from the pathogen. And to simulate such a mechanism, we use two different approaches. The first one is based on PDE, so partial differential equations. So we have a three-dimensional environment with the molecules that we considered as concentrations that diffuse on a discrete grid according to the gradients. So here is how the model looks like, and we have the different parameters such as the secretion rate of defensive molecules by the pathogen, the uptake rate of this antimicrobial peptide, the diffusion coefficient of the molecules, and also parameters related to the complex dynamics. And as a readout, we can look at the time-resolved spatial distributions of the molecules. So here we have on the y-axis the concentration of our three different molecules, and on the x-axis the distance to this pathogenic cell. So at the initial state, we have a homogeneously distributed antimicrobial peptide, and no complex nor defensive molecules. And what we can see is that in red we first have the secretion of defensive molecules, and then these defensive molecules interact with the antimicrobial peptide, forming complex imprint. And in this way the concentration of AMP is reduced in the vicinity of the cell. And another thing we can look at is a so-called score, which is basically just the number of antimicrobial peptides that have been uptaken by the pathogen in regards to the initial number of AMP that we had in the system. So here, bright orange means that almost all the antimicrobial peptides have been uptaken by the pathogen, which is bad for the pathogen. And blue means that we have this beneficial regime for the pathogenic cell, this immune evasive regime, because we have a high reduction of the uptake of AMP. And so we computed this score for, we screened over 4 different parameters, so the association and dissociation rates, uptake rate, and secretion rate. And we observed this beneficial regime for the pathogen for quite a wide range of parameters, and especially when we have a high secretion rate and a low unbinding rates. So this was for the PDE approach, and we also used an agent-based modeling approach. So we again have our three-dimensional environment, but the difference is that now we consider the molecules as single agents that perform a Brownian motion. So here is how it looks like, and we have again the same interactions as in the PDE. And when we look at the time result spatial distributions of the molecules, we can see similar dynamics as in the PDE model. So we have first in red the secretion of the defensive molecule in the vicinity of the cell, on the cell surface, sorry. And then the formation of complex in green, and this results in a lower concentration of antimicrobial peptide in the vicinity of the pathogenic cell. So as a conclusion, we can see that the secretion of molecules by the pathogen reduces the concentration of antimicrobial peptide in its vicinity, and then thus increase its chance of survival. And this was shown in both approaches, so in both the ABM and the PDE approach. And such a mechanism is also supported in the literature, because it has been shown that Candidalbi can secrete MSB2, which is a molecule that shows high affinity with LL37, which is a human antimicrobial peptide. Thank you.